I now have the honor of introducing Fran Grant and Mark Arnholt. Fran and Mark have almost 50 years of teaching experience between them, and they both teach in the Project Lead the Way biomedical track program. And then actually they live within a few blocks of each other here in Wisconsin. They have attended numerous CBM and Project Lead the Way summer courses on the MSOE campus. Fran Grant teaches at Grafton High School, where she's also the Future Health Professionals Advisor. Fran earned her bachelor's degree in biology from UW-Eau Claire and her master's degree in curriculum and instruction from Concordia University. Mark Arnholt teaches at Hartford Union High School, and he is the SMART and now MAPS team advisor. Mark's teams have presented their projects at national conferences, and one team earned first place in the CBM's 2016 SMART Team Symposium. And now with that, Fran and Mark will begin with A Tale of Two Sisters, Modeling Transcription and Protein Synthesis. Take it away.
Hi, everybody. Thank you so much for the wonderful introduction. My name is Mark Arnholt, and I guess to start off here tonight, we need to kind of get some normal things out of the way first. Like, you're going to need space today. Like, if you don't have space in front of you right now, like, arrange things so that you do have enough space to actually work. You're going to want to have your student modeling pack out with the RNA polymerase out. You're going to want to have your DNA nucleotides, and the DNA nucleotides are a little bit different than the RNA nucleotides. So the ones that are round on the bottom are the ones you want to start with. And you're also going to want to have another device kind of ready so that you can actually watch the presentation here, but you can also interact with us with your Pear Deck. So if you've got your cell phones handy, like, get those out, get them ready to go. And you're going to want to have your browser open on that particular, on your device, right? The other thing is that some general norms, like don't be afraid to ask questions. Like, questions are how we learn. Love having questions and interacting with the audience. And feel free to, like, use those chat options to put those things in. We do have a DNA sequence we want you guys to have put together. And again, when you're using those, you want to make sure you're using the ones that have the round bottoms. These are the DNA nucleotides, and there's a distinct difference between the DNA and the RNA. So have those ready to go. The RNA nucleotides are square, and we're going to be using those a little bit later. But right now, just have the DNA ones out and ready to go.
So I am sure at this point in the school year, most of you are familiar with Pear Deck. Well, when this presentation was first put together, Pear Deck was fairly new to me because this was probably last March. And I find it really helpful, especially with my virtual students, to use this as a way to get some sort of interaction going, because I'm teaching in a situation where I'm asynchronous. So I have students in the classroom and I also have students online. So to try to make this presentation a little bit more interactive, like what I'm trying to do with my virtual students, I'd like us all to go ahead and join Pear Deck. And we'll be using Pear Deck throughout the presentation as a way to, again, create some sort of community. So all that you do is, using a cell phone or another device, you just go to joinpd.com, and then you can see on the screen the code that you need to enter. And you'll also be able to see the slides as we go through the presentation. And you'll be able to respond on certain slides that have been set up to use Pear Deck.
The other thing we want to have you guys put together is your first genetic sequence. So we're only doing one strand, and we're starting off with CCG. So I know it's hard to, like, read the nucleotides right now, which is why it's beautiful that they're color coded, so that you can literally just build it that way. But this is the sequence that we're going to be working with tonight. And if you don't have it, like, ready to go right now, I know that they just posted it in the chat, so you'll be able to kind of have that up and underneath. while we're actually starting to go through some of the presentation, you can build that. I know that like you'll have your mind actually trying to occupy two spaces at the same time, but it'll be okay.
Yes, it will be. We have some filler. Like, for example, I would like to just share with you while you're putting your pieces together, the story of Helen Obando. But before we show the video, I just want to let you know that as a biology teacher, I think we have probably all shared a lot of scenarios with our students about other patients or other people that have had sickle cell anemia. I find this one about the story of Helen Obando just fascinating for so many reasons. Keeping in mind that I'm a PLTW biomedical science teacher, and I do teach medical interventions, which is the 3rd year. And in the 3rd year of that course, in Unit 2, that is where our students study reproductive technology. And in that unit, we address gene therapy, but we also address pre-implantation genetic testing. It's abbreviated PGT. And that is also part of this whole story. And I'll get into that in a little bit once you watch the trailer of this video. But this scenario is from the New York Times. It was published January 11th of 2020, so just about a year ago. And there is a full documentary on Hulu under The Weekly is the name of the documentary series on Hulu. So take a look at the trailer to get introduced to Helen Obando.
Sometimes it just comes out of nowhere. Pain will start in my arms, then it will go to my legs, and it'll start in my back. I describe it as needles poking me all the time. My sweet 16, my friends that were there, they didn't know I was in pain. They wanted me to like dance, have fun, and all of that. I've gotten used to like showing that, oh, I don't have pain. I'll just act like if I'm normal. I never thought there was going to be a cure. I always thought I was going to stay with sickle cell like forever. This trial, no other teenager have gone through it yet. I'm going to be the first one.
So just a little bit more about the story. It's a long story. Like I said, the documentary is about 40 minutes. But this is about a Hispanic family that is unique in many ways in that they have 3 children. Their oldest daughter, Haley, also has sickle cell anemia. Helen is the middle child. She has sickle cell anemia. And then they have a son named Ryan, and Ryan does not have sickle cell anemia. Now, this young couple, I mean, what a nightmare to be going through. They were married very young. What they did find out after Ryan was born is that he wasn't able to be a donor for Helen. He was able to be a donor, a marrow donor to the older sister, Haley. So Haley was cured by having a bone marrow transplant, or she is in remission, I should say. Unfortunately, Helen's symptoms were much, much worse, and I'll talk about her symptoms later. And Ryan was not a match for Helen. And so the parents, Mr. and Mrs. Obando, actually went through they wanted to be able to see if they could have another child that would be a match for Helen. Now, Mr. Obando had had a vasectomy, so there was that whole issue. So they had to get sperm from him. They had to put Mrs. Obando on several drug therapies so that she would release a lot of eggs at one time. Through in vitro, they actually were able to create a lot of embryos, and they went through 73 embryos, and none of them were a match for Helen. they spent $90,000 in this process. And so you can see it just brings this whole story of sickle cell anemia into so many levels, how complicated it can be to have children with a disease. And I do have to give, so Helen Obando is in a trial that is, has been put together by Dr. David Williams at Boston Children's Hospital. And so that is my intro to Helen.
So part of this is to kind of give you guys how to utilize the flow genetic information kit in your classroom, and also to tie in or show you some of the next generation science standards as they relate. The other big component is to not only give your students who are in person a hands-on experience, but also for the students who are actually attending classes virtually. And then hopefully give you something interesting and new as far as professional development goes, and also hopefully give you something to bring back to your classroom almost immediately.
So we would like to know who's here. I have heard, I haven't seen, but I've heard there are over 150 of you out there. I can't see you really, but this is one way that I can at least, or Mark and I can get an idea of who we're talking to. So we'd like you to use your Pear Deck, and it's a multiple choice example. And we just like to know who we're talking to, what it is that you teach. And this is great for your students. So your students won't see the results right away, but you will have a teacher screen if you use Pear Deck as a presenter, and this is what you'll see. You can also hover your mouse over the teacher screen and you'll actually see your student names. So we're using this as an example to get an idea of who's here tonight, but you can also use this as a formative assessment tool with your students that are virtual and in person. So I see we have a lot of bio people here, Mark.
Beautiful,
a lot of AP bio people here.
So the next question, of course, is why use modeling? And a lot of times when we start getting into, especially the more abstract concepts, that this gives those students a hands-on way to start wrapping their brains around some pretty difficult biochemistry. And so there's lots and lots of different models that we can utilize. And again, Fran and I are both PLTW teachers. So that stands for Project Lead the Way. And we are in the biomedical sciences track. So as we go through things, a lot of times we're always linking them back to specific biomedical science standards. And so like this is the shorthand for us. PBS is Principles of Biomedical Science, and the things that students in the first year of the track would actually do. Year 2 is Human Body Systems, or HBS, and some of the learning objectives we have for them. And then MI is year 3, which is Medical Interventions, which is the third year. And there's actually a 4th year on top of this, Biomedical Innovation. And that one's actually a lot more on problem solving. And like I actually brought him out this year when we were starting to talk a little bit about CRISPR and Cas9. And so like there are multiple places you can use this if you're teaching biomedical science. I know most of you probably aren't doing that, but you are using the next generation science standards. So we've actually linked this back to those, including the Science and Engineering Practices, or SCPs, the cross-cutting concepts, and then the disciplinary core ideas. So if you're trying to find a way to promote these, or you have to like justify using these to, I don't know, your director of curriculum instruction, like here are literally the exact learning targets that they actually meet.
So back to Helen. So like I was just saying, of the 3 children, Helen had the worst symptoms. So at 9 months old, she ended up having a blood clot in her pelvis. And you can imagine the pain that a 9-month-old child must have felt. So when I watched this documentary and I was reading her medical history, I just felt just horrible. I mean, just horrified by her experience and what she must have gone through. At 2 years old, she was hospitalized to have her spleen removed. She had a clot in her spleen. In her young life, she's 17 now. She was hospitalized 13 times with clots in her lung, acute chest syndrome. She's had bacterial infections in her spine that she's had to be treated for. Her pain episodes, as you saw in the trailer, are excruciating. One of the things that you'll hear, you know, after she has gone through her treatment, she actually has sore muscles because she's able to do some physical activity, and she didn't understand what sore muscles felt like, because the only pain she's ever known in her life is from sickle cell crises. The other thing that I like about this story is, in most of my career, when I've addressed sickle cell anemia, it's often been with an African American individual. And I think it's important for our students to understand that sickle cell anemia isn't just a African American disease or a Black disease. It's a disease that affects a lot of different races. So one thing that you can do, so, you know, when I'm starting, it's funny, I'm starting this unit today or tomorrow. I'm showing the video tomorrow to my, my third year at medical intervention students. But one of the things that you can do, and you can see on your device, is that you have this red dot. And what I would say to my students is, look at these two blood smears. Can you draw your dot to which blood smear belongs to Helen Obando? And I'd like you to do that now. You can see the dot, you can drag it, and you can see how you could use this in a lot of different situations. And so all of you have a strong biology background. So you all are drawing your dot or moving your dot onto the correct blood smear. But again, as a teacher, you can actually hover your mouse on the teacher screen over the dots, and the name of the student will show up. And it is fun. You do get students wiggling around the dots and they get all excited about it.
So another one of the things that we're kind of starting to do is, like, this could be like a whole unit or a mini unit. So like, this isn't something you're probably going to cover in one day. This is something that's going to take you multiple days to get through. And there are lots and lots of student modeling packs that you can actually utilize. In the one that you have now, you already can see that you have something for transcription. So you have the RNA polymerase, but you also have the DNA polymerase and the DNA helicase in this kit as well. So there are lots and lots of different tools that you can utilize to actually start building together a complete unit that all deals with some of these molecular biology concepts. As we look at this, there are other models as well, including, this is actually one of the old DNA models, and I can tell that it's an old one because it doesn't actually have the dynamic abilities that the new one has. Like, the new one, I can actually untwist and I can actually separate into individual strands. And the purple thing that's wrapping its way through it is actually an amino acid starter kit. So you can start talking a little bit about how DNA can actually be recognized by specific amino acid sequences and specific protein shapes. And this is actually two like short videos that kind of show everything in kind of sped up. The one on the left is for the DNA replication. And before I started doing things on transcription and translation, I actually would have spent time covering this with my students. We've already would have covered basic DNA structure, and we would have talked about how DNA is actually replicated. And today, the big thing that we want to focus in on is the actual process of transcription. So how do we get our DNA copied into an RNA strand, and then how do we get that RNA strand out to a ribosome to actually start doing the job? This is also a really good time to kind of ask students, well, like where in the cell are we? Like what's going on? Where are these components actually located? And so I know that many of you have probably taught DNA structure before, and here's like the basics behind a DNA nucleotide. So we've got our phosphate group on the far left side. We've got our deoxyribose in the center. and you probably spent time saying, oh, start at the O and then count going clockwise and number those carbons. I've got carbon 1, carbon 2, carbon 3, carbon 4, and then up top I have carbon 5. And then off to the right-hand side of this, we're actually going to have our nitrogenous base, whether it's an A, T, C, or G. As you look at your individual nucleotides, you'll see that some of those same things have been put in, and that we've actually demonstrated the 5' side and the 3' side. So the 5' side is the side that actually has like the receptacle in it, and the 3' side is the arrow side on the other side. And then coming up off the top, you actually have each individual nitrogenous base. So you actually spend time just talking about this. You guys start talking about how we form phosphodiester bonds and talk about dehydration synthesis. And you can also use these to actually start going into some of the intricacies of the bonding that happens between bases. So this is a point in time where, like, just because of the model, like, you're going to start having people ask questions. So the first question I ask you is, if you need to replicate the strand, you need to make the complementary strand to your DNA, like, which side should you start on? Like, where's the starting side to actually replicate the DNA? And as I was doing this, I actually had my children assisting me as I went through this. And my daughter on the in this photo is actually a student who's going into pharmacology. She's a third-year pharmacology student. And my son is actually going into real estate. So I said, all right, well, which side do you start on? Because the second I said replicate the strand, they started just building from both sides. And, well, interesting outcomes. Who's starting on the right side?
Yes. I am.
Now, mind you, this is a third-year pharmacology student. She's gone through biochemistry, and like, these are things you just think they naturally know. But again, a lot of times
we're going to have exactly enough A's.
They start to have these realizations that are happening while they're doing this, and like, oh, like I have to start on that side. I can't start on this side because the DNA is always going to be copied from the, well, it's going to be built in the 5' to 3' direction. So starting point, what you guys need to do is start building your complementary strand of DNA.
And that brings us to another Pear Deck slide. So, and again, you all have a really strong biology background, but this is, again, we're just, wanting to show you what you can do with your students. You can have them draw a dot and just say, please place a red dot where you think the template strand is located. And everybody's just wiggling the dots all over. This is just like school. Very good. I think we all got it, right, Mark? Does it look like we have it?
So another one of the things that you're probably going to have talked to your students about already is some of the big differences we have between DNA and RNA. So we say DNA and we just kind of to go deoxyribose. So what does that mean? Well, as we're looking, we go down to that 2' carbon, and we notice that it's actually missing that oxygen molecule there. Whereas if we just look at the regular ribose, like we've got a full hydroxide group there. So we've actually represented that by making a difference in the nucleotides. And I always love, like, when I get to the end of the unit, I'm like, all right, so what have we learned about like the differences between DNA and RNA? And the students go, RNA is square. And you're like, no, that's not.
I know.
What's happening? Right.
I know.
But a lot of times, like, they're bound to the model. So again, this goes back to that idea that all models are wrong, but some models are useful. So every model has limitations, and it's the time to begin thinking a little bit about what it is exactly that like, how can I utilize these? What's great about them? What are some of the flaws inside of it? So that ends up always being one of the things that I'm like, oh, like, please. There's other things to look at here, right? So we've got uracil, which is actually replacing our thymine. So we don't have any T's inside our RNA. And then we've got square ones versus round ones for our modeling purposes. So once you actually have your DNA built and you've got your full strand, all right? And again, like, this in and of itself is a powerful tool. Like, you can start utilizing this to start talking about purines versus pyrimidines. You could use this to talk about hydrogen bonding inside of it. Or if you want to get a little risky, you can start talking about how hydrophobic interactions are playing a role in holding these things together, all right? Lots of things that you can actually do. You can also use those arrows to show how those phosphodiester bonds are actually stronger than the hydrogen bonds that we have that are actually holding the two strands together. So there's lots of different things that you can use to actually start adding depth of knowledge to their basic idea or foundations on DNA. Anyway, what you need to start doing is actually start slowly taking your DNA and start wedging it in against the RNA polymerase, all right? And as you're doing this, like I already know you guys, you already punched out all your things and you've already probably tossed out the extra foam. But I would suggest maybe keeping that piece or some of those pieces of extra foam and then cutting them so you can actually place them so you actually have the wedging action that happens when you're actually utilizing the classroom packs. In my classroom, like I'm hybrid right now, so I actually have some students who are showing up in person and some students that are doing these things from home. And I want to make sure that they're all having similar experiences, all right? So as you're working that in, you're going to wedge that DNA apart, and slowly and steadily, you're going to have those nucleotides arrive in the active site. And every time that one of those nitrogenous bases from our template strand hits the active site, we have to bring in the corresponding base pair of RNA. And slowly and steadily, that RNA is going to build its way out over that bridge that you've installed, and you'll kick the RNA out through that pore kind of on the bottom of your model, all right? What you'll need to do though, is as you're doing this, is slowly and steadily show that that DNA is conserved, like nothing happens to it, it doesn't change, it's just being read to make that messenger RNA to send out to the ribosome. So those nucleotides come back together after they've been through the active site and form DNA again. So the DNA nothing happens to the DNA, it stays exactly the same. We build that RNA strand that's going to actually come out through the pore. And now we have our messenger RNA to send out to the ribosome. So now we've got to leave the nucleus to go do that. So there's our, one of our things. The other thing to point out is that, hey, that strand is exactly the same as that bottom strand was, except now every place that there was a T, we now have a U, all right? This is actually utilizing the classroom kits. And this is something that I would normally do with like a group of like 4 students. So as they're doing this, a lot of times I kind of give them jobs, and as part of my assessment, I usually have them make videos. So they record these in real time and kind of have to go back through and narrate them to kind of hold them accountable for the content that's actually happening. But what I really love about the classroom kit is that it has the foam on the mat and that it naturally wedges the DNA apart when it actually enters. And it's also got, it actually funnels it back together. So it's naturally going to bind back together on the back side to make your RNA strand.
And it's much bigger too than what a lot of you have in front of you. If you don't have the classroom kit, this will take up a full lab table, wouldn't you say, Mark? Yeah.
Like they're big, like they're like,
yep.
There's real size for one of these. So. One of the other things I love about this is as I use them, like a lot of times, like they end up using it as freshmen, and then I get them in MI or BI, and I pull these out, and they're like, oh, like we've done this already. And there are so many layers of complexity you can actually add on top of just the natural kit, right? And one of the things you kind of have to be ready for is the fact that this is naturally and organically has students start asking questions. And that's the beauty of it. Like a lot of times, one of my favorite quotes, the problem with education is that students are rewarded for knowing the answers to questions they've never asked. Using these models forces them to start asking those questions, and it's just one of the natural things that happens as a result of using them. So as this was happening, again, I was recording my 2 kids at home working with these models. And here's examples of some of the types of questions you may need to address or be ready for as you're using these in class.
Okay, so now we've gotten to a T. Do I do an A there? And then the A, we do the U.
So now you gotta keep sliding it though. So just some very, very simple processing, right? Like, oh, like we're in RNA now. So, so A is gonna bond to U, but T's are still gonna bond to A's. So A's are something that's conserved, but the U's are something that are different. This is like a really, really great question that has so many different answers to actually address or different directions to take it
in. So where does the RNA go? Because we're like running out of space.
So, well, again, what happens with DNA? Where does the RNA go? We're running out of space. Oh, okay, well, how do we want to address this? Do we want to start talking about the fact that there's a pore where the RNA comes out and it's just gonna leave the nucleus? Do we want to start talking about, oh, well, hey, before it leaves the nucleus, there's other things that have to happen. We have to put our poly A tail or have polyadenylation happen. We have to have a methyl cap put on the other end, and that this is one of the signals that actually tells it to leave the nucleus. Or you can go to the next level where we start talking about alternate splicing and spliceosomes and splicing factors and how we can actually get one gene that codes for lots and lots of different proteins. We take these messenger RNA pieces, we cut them up, we switch them into different spots, put them back together, and we get a different amino acid sequence. And then we polyadenylate, and then we methylate, and then it leaves the nucleus to go to a ribosome. So there's lots of different ways that you can utilize this and lots of different layers of depth you can add to something that they're like, oh, we've done this before. I'm like, oh, well, let's talk about something else and add another layer of complexity. Fran, you got to unmute yourself.
Oopsies. I was just going to thank you, Mark. I was going to say, or you could just be real simple and just draw a line. And this again, this is a David Goodsell image, and 3D has a lot of David Goodsell's work, and it's just beautiful. They also, I have them. Mark has a couple of images behind him there, but this is an image that's provided in the teacher resources with the Flow of Genetic Information kit. And I think many of you have probably already drawn an arrow to help address that question. Well, where does the mRNA go? And so the nucleus, as you can see, is this area that's in yellow. You can see the nuclear membrane and the nuclear pore. And then you can also see where the ribosome would possibly be, but it looks like here you can just kind of go out into the cytoplasm. So let's see what you guys all drew. Oh, see, now that's neat. I didn't know that everybody would get a separate slide. I thought, I've never seen that before. It's usually a whole bunch of drawing on just one slide, but that's very cool. And so that's just another way to get your students to think, well, what happens to this mRNA?
It's also a great way to get immediate feedback and a formative assessment.
Yes.
So now we've made our messenger RNA. What happens to it? Well, okay, we've got a bunch of different things that can happen to it for it to leave the nucleus, but now we're at the point where we need to actually have the translation component happen. So in the classroom kits, there's actually another mat for translation, and it actually shows the two different ribosomal subunits. You can actually take your messenger RNA and start working its way through. You've got transfer RNAs that can come in. You can actually have or start having that discussion with your students about codons in the messenger RNA and the anticodons on the tRNA and how they'll bind together, and how every time that happens, we bring in another amino acid which gets dropped off, and we start having the elongation of that protein begin, and we're getting that primary structure. And again, a lot of times I actually have my students like I hold my students accountable, not by giving them a pen and paper test, but by having them put together short videos and then do explanations of them. So they actually have to narrate over the top. And there are specific things inside my rubric, like you have to talk about the active sites. You have to talk about codons and anticodons. You have to talk about how the amino acids are linked together. So there's lots and lots of different things you can do this besides fill in the circle and pick the right one. This is also a point in time where I make a plug for the next kit, which is the amino acid starter kit, another one that I absolutely love, which you can actually start utilizing to show some of those ideas or concepts behind protein folding.
Mark, I think this is an example of one of your students, right?
This is. So I actually got permission from one of my students last year to show a little clip of their video.
When the DNA is split to get RNA, it's split through an RNA polymerase. Once this happens, the top side of the split DNA is matched up with the right corresponding RNA nucleotide. The only difference is that the T changes to a U in RNA. The pairs go like this: T to A to U, C to G, and G to C.
So you can start to get a feel for the types of things that you can actually do and the types of things that your students can do. The other thing I want to talk about is the fact that, like, I've done it as a stop motion before, where they take individual pictures and then, hey, move it over one spot, like take another individual picture. And unless you want to spend like a week where they're just making a video, like just have them record and do it in real time and then come back over the top and narrate. And like WeVideo is a wonderful, wonderful free program you can use for your students to actually have them start doing some of this video and video editing.
So now that your students have background in transcription and translation, and we actually have our mRNA that was read by the ribosome, well, now what? So how did these sickle cells actually happen? And this is where you can get into that lesson about genetic mutations and about a single base change that is the cause of sickle cell anemia. So I know most of you already know what that mutation is, but now what you can do is expand upon what they've just learned. So now we have created this mRNA, and we have created from that mRNA, we have created this sequence that has valine instead of glutamic acid. But then they have to understand, well, what does that do? And that's where the amino acid starter kit, I know this presentation isn't on the amino acid starter kit, but the amino acid starter kit allows students to actually fold the protein into its shape. And with this story, what I love about this story is that this is a slide of adult hemoglobin. And adult hemoglobin does not have as strong of an affinity of a molecule called 2,3-bifoglycerate. And so this is what actually is affected by this base mutation that was just on the previous slide. And the specific gene that we're talking about is the BLC2A gene. Now, Dr. Herman is going to be talking more about this following our presentation, but the treatment, the gene therapy that Helen Obando received is a treatment, it's a form of gene therapy in which bone marrow cells or stem cells were taken from her, taken out of her body, and using the AIDS virus actually delivered, now this is what we're trying to, Dr. Herman and I were talking before the presentation, it delivered a specific, what we're trying to figure out is whether it knocked out the gene, the B part of the BCL2A gene, but the AIDS virus nonetheless delivered a gene or a molecule that would actually switch off the gene that creates adult hemoglobin. So therefore, in Helen's body, fetal hemoglobin was being produced instead. So once the cells were infused with this gene therapy, those cells were then put back into Helen's body. Now, granted, this was a grueling process for her. She had to receive chemotherapy to actually kill the bone cell, her bone marrow. Then she received her cells that were now infused with the gene that produces fetal hemoglobin, and the gene that makes adult hemoglobin was actually shut off. And so this was infused into her. And it is amazing. The results within a year, what we have seen. And this is, by the way, a picture of the adult. We can go back, Chris, but this is the picture of the amino acid starter kit. Right here, yeah. And so, like I said, you could see this. These are my students. They're actually folding a protein based on the characteristics of the amino acids. And so that's something you can do with your students so they understand, well, what's the big deal if we just have this one base change? But then an update. So I will tell you that Helen Obando was the first teenager that was treated with this form of gene therapy. And she is doing fantastic. After about 6 months after gene therapy, she was actually able to be outside. This took place in Boston. And since this time, the family has now moved to Mesa, Arizona. But when she was able to leave the hospital, she was able to actually be outside in the cold, where before her gene therapy, the cold weather would bring on these sickle cell crises. So she really had to be careful in cold temperatures. She is now dancing. And like I was telling you earlier in the presentation, she has sore muscles from dancing. If you watch the documentary, you see her, they went zip gliding. She's very active, where before she wasn't able to be very active because she was in pain. She didn't have a lot of friends because she would have to go home from school a lot. She was in the hospital a lot. She is just like every other teenager. She's out there on social media. These are pictures from her YouTube channel. Now, I did friend her on Facebook, but I am not a friend of hers anymore. And she hasn't really posted much since Thanksgiving, but she is beautiful. She has long blonde hair right now, and she strikes a pose like all of our students do. I'm sure she's on TikTok. I have not found her on TikTok. I don't have a TikTok. But she does have, like I said, a YouTube channel. She hasn't really posted a lot. She posts videos on how to put on makeup, how to do your nails, that type of thing. But it's amazing how her hemoglobin levels are normal, and she does not have any evidence of sickled cells in her blood at this point. So it's an amazing success story to share with your classes.
Thank you, Fran and Mark. Oh, sorry, I'm on my wrong video. There we go. So thank you for that. And we have time for questions now. There was one that you guys mentioned a whole lot of things during your session that people wanted to have links to or information for. And so I will be collecting all of those links, and I will be sending up a follow-up email with the link to the video and all of that probably on Friday. So you will get all of this and the slides and things. And Mark was talking about adding an extra piece of foam. And so let me go ahead and show that to you now. And if you have other questions for Mark and Fran, or if you have other ideas and things that you have done to with either the nucleotide student modeling pack or the FlowJack information kit, we would love to get your comments or questions right now. So this is the part that came when your nucleotide student modeling pack, and I don't have the frame by me now, but I have another piece of red foam. And so what Mark was saying is if you would put a piece of red foam right here on this arrow part, then when you put your DNA through here, and then it will actually separate a whole lot easier. So a much smaller piece would work better here. I just don't want to. I'll need the substrate later for another session. So that is a great option for you guys if you want to do this. And there were some other questions about, you know, the difference between our student modeling packs. And this is really what we created just for this year so that students would be able to have those same modeling experiences either at home or individually with no shared materials in school. Our full, you know, this is the pared down version of the flow genetic information kit of one of the mats that come in here of transcription. So you can see typically a group of 4 students would be working and doing this modeling with longer sequences and things. But this is something that at least gives the students some hands-on modeling during the school year. Okay, some other questions have popped up. So Karen, you were asking, sorry, questions are a little far away from me. Does somebody want to jump in and ask?
Heather, one of the questions that Karen asked was the reason they needed to do that, was the reason they needed to do that for this girl? I'm not sure I understand that question. Karen, if you can, was there a reason? I'm gonna move down, Karen. I'm gonna keep looking through to see if I can follow your question. Rachel asks, she has the classroom group kits and she sees there are different sequences. Are there any special ways you use these sequences or randomly assign them to students?
The big thing is whether you're planning on doing both the transcription and the translation. Like if you've got a block session, like you actually have sequence 1 and sequence 2, and it actually tells you on the bottom whether you should use this for like replication and transcription, or if you actually want to do one for transcription and translation. So you actually, with the modeling kits, sorry, don't mind me. I just had to run over and grab it quick.
That's what I was going to say. There's a PDF also.
Big long sequences to actually utilize. And on the ends, it actually tells you, like, are you doing replication? Are you doing transcription? Are you doing translation? And the reason for that is because when you're using the translation kit, that there are some limitations to it because there's so many different codons you can have and bring in so many different amino acids. So in order for it to work, you have to use the right DNA sequence to go from DNA to RNA, and then to go from the RNA to the actual amino acid sequence. But that's a great question.
Okay, there are lots of questions here. So, and I see Mary, you are looking for some recommendations. Oh, that your online students would be able to do something even if they don't have like the student modeling pack. We do have digital drag and drop slides associated with all of our student modeling packs as well. So they would be able to do at least some modeling associated with that. And I
actually had some students doing that with replication because they didn't come in to pick up the student modeling pack. So I was like, all right, well, if you don't have the modeling pack that I got for you, like you can do this online version. So the CBM actually has lots and lots of, 3DMD actually has lots and lots of really, really wonderful resources for your digital learners, especially if they can't actually make it to like pick up materials.
Some of the questions.
I think I understand what Karen's asking. She had asked a previous question about whether or not they needed to turn off the adult hemoglobin, and Tim answered no. And so I think she's looking for more clarification, like was there a reason they needed, what was the reason they needed to turn it off? I don't know if we can hold that question until after Tim's presentation. That might answer that question a little bit more thoroughly.
Otherwise, what I was going to say is because the mutation, fetal hemoglobin isn't susceptible to being sickle celled like adult hemoglobin is. And I know Tim can explain more about why that is. But because usually when we're born, I see Jan just sort of, when we're born, the fetal hemoglobin stops being produced and the adult hemoglobin starts to kick in. But the mutation doesn't affect, the sickle cell mutation doesn't affect fetal hemoglobin.
Okay, and we are back. So now I would like to introduce Dr. Tim Herman. He is the founder and director of the MSOE Center for Biomolecular Modeling and the founding partner for 3D Molecular Designs. Telling protein stories is one of his favorite pastimes, and he is enjoying the opportunity to make videos of his favorite models and stories right now. Please subscribe to the 3DMD and the CBM YouTube channels to catch all of his videos. And now here's Tim.
It's always interesting for me to see how teachers can connect some of these basic things like transcription and translation to the story. And I'll just have to say that Obando story, you really need to make the effort to watch that video that's on the New York Times site. It's a long video, but man, is it ever powerful. And if there's a way you can get your students to see that, you're going to have them really hooked as to this story. So, and that session raised some great questions, which I hope we can address here in the next 15 to 20 minutes. So I'm going to talk about the first molecular disease, which in fact was sickle cell anemia. Way back in 1949, we'll talk about the guy who first showed the molecular basis for sickle cell anemia. And before that, I'll talk a little bit about the structure of the hemoglobin protein. So let me start with that. So I have in my hands here a terrific model of hemoglobin. And I'm not going to spend much time because I think everyone is familiar with this. Hemoglobin is a heterotetramer, meaning there are 2 red beta globin chains and then 2 white alpha globin chains. And on this terrific little model, I can take the beta globin chain off. So it's magnet docked onto the other 3. So the sickle cell mutation occurs in the gene for the beta globin protein. So that's why we're focused on it. And on this model, you can reach right in here and you can pull out the heme group. So the heme group is this little small molecule. And on this heme group, there should be a molecular oxygen, but I can see right now that it's not present on this model.
Grabbed the wrong one.
Yeah, but that's okay. I'm going to switch right away then to this bigger model of the beta globin protein. And I'll tell you that you should watch for these bigger models because we've recently developed this technology, become familiar with this technology. We didn't develop it. But this is made on a low-cost 3D printer, and it's made of plastic. It's not fragile like some other models. So, you know, we used to create these mini models made of plaster. They're wonderful, but if you drop them on a hard surface, they'll break, and they're kind of small. So what I'm showing you here is that in the not too distant future, you're going to start to see larger models made of harder plastic. So here I'm going to pull this heme group out, and in the middle of that heme group there is a orange oxygen, and I can take molecular oxygen. What did I say? In the middle of this heme group there is a iron atom. That's what I meant to say. And you can then take a molecular oxygen.
Okay, Hold that closer to the screen.
Hold that closer
To the camera.
screen. Up here?
Yes.
Aha.
Oh,
Okay. It's out of focus.
there we go.
Okay. Yeah, we're still becoming familiar with all of our setup here, but there is molecular oxygen bound to iron. And we sometimes call this a roadkill toad. You see his two back legs here with his toenails. So he dives into this hydrophobic pocket right here with the polar toenails sticking out. And there are two histidines on either side of that iron atom. So that's how the heme group is bound. And then let's go over here. Let's find the N-terminal end, which is right here. And if you count in 6 amino acids, you will find a glutamic acid side chain in normal beta globin. So let me see if I can orient that. Right here is glutamic acid at position number 6. Then the problem then with sickle cell anemia is that a person with sickle cell anemia has a 1 base change in the gene that encodes this adult beta globin protein, such that the glutamic acid codon was changed to a codon for valine. And valine is a hydrophobic amino acid, whereas the glutamic acid is a negatively charged amino acid. So when you have glutamic acid there charged on the outside surface of beta globin, then that contributes to the solubility of this protein, and in fact, the solubility of the entire hemoglobin tetramer. So you can have high concentrations of this protein inside a red blood cell, and it's interacting with the water that's in the red blood cell, and it's soluble. But if you then change this charged amino acid for a hydrophobic amino acid valine, that small molecular change there is what results in sickle cell anemia. You put this hydrophobic patch on the outside of a hemoglobin, tetrameric hemoglobin. And the next thing that's going to happen then is that these hemoglobins are going to start to aggregate. So the hydrophobic patches that is introduced cause the hemoglobins to stick together. They form these long fibers inside of a red blood cell. And those fibers push on the inside surface of the red blood cell membrane. And they cause that distortion in the shape. They make a sickled cell red blood cell. And then those sickled cells start piling up in capillary beds, cutting off the blood supply to certain parts of the body. And that's what results in these really excruciating pain crises that many sickle cell patients experience. So
I'm going to interrupt you here for just a second, because this is something that I have learned that a lot of teachers have misconceptions about. So let's repeat this again. The mutation for the sickle cell does not result in a significant change in the shape of the protein.
That's
correct?
True. That is correct. And I'm looking here. Boy, we're missing a few side chains on our model. So yeah, here we are right here. So here's a glutamic acid. And in many ways, it's too bad that we always focus on sickle cell anemia and hemoglobin, because in many ways it doesn't do what we would like it to do, so that we can make this strong message lesson for our kids. Because we teach that a change in the gene causes a change in the amino acid sequence, which causes a change in the structure of the protein, which inactivates it. This doesn't cause a dramatic shift in the shape of the protein. So that's why we don't have a beta globin, a normal beta globin protein, and a sickle beta globin, because they're really not much different in shape other than this side chain right here. So it is true that there's not a dramatic conformational change. There's something else. There is a shift in conformations here, but it's something we really don't have the time to talk about right now. And that's a shift between the oxygenated and deoxygenated hemoglobin. But yeah, we don't have time to do that right now. So we want to focus on sickle cell anemia. And I want to move on to this second slide then, which this is the first molecular disease, right? So this is what they're seeing now. Is that
Yes,
slide?
it is.
Okay. So this was discovered, reported in a paper published in 1949 by a guy named Vernon Ingram. And Vernon Ingram was working with a famous chemist, Linus Pauling, which we probably all heard of. And Vernon Ingram, here's some data from the classic experiment that he published. They knew at that time that there was a difference in the blood of a person who had sickle cell anemia and the blood from a normal individual, healthy individual. So they could look at those red blood cells under a microscope, and I think Fran showed a slide like this. Why can't find my mouse. There it is. Okay. So you can see a couple of really nice biconcave red blood cells up here in this micrograph, but you also see these sickle cells. So they knew there was a difference in the blood, and then they knew that these red blood cells were chock full of hemoglobin protein. So the hypothesis was there must be something different about the beta globin protein in the RBCs, the red blood cells from a normal person versus someone with sickle cell anemia. So Vernon set out to prove that. So his approach was to take blood from either a normal individual here on the left or this person with sickle cell anemia, and he digested the protein up into pieces using a protease. So he used a protease that cut only after basic amino acids. And I'm telling kind of a sloppy story here. I don't know exactly how many peptides you get when you digest beta globin, but let's say you get 10 or 12 different peptides. So he did this for normal blood. He did this with sickle cell blood. And then he ran those peptides out, not just in one dimension on a gel. This wasn't a gel at that time. It was a layer of starch. So he did some electrophoresis in not only one dimension, but two dimensions. So he got this two-dimensional pattern of peptides from these two samples of blood. And if I let you stare at these two, I'm not sure what you call these, electro something grams, you would eventually see that there's a difference. And I'm going to ruin it for you now. If you haven't already seen it, you see this little faint peptide right here? It's not present over here. So this was the whole basis for the first molecular disease, because he eluted this peptide out of this starch plate, and he determined the amino acid sequence that was in this peptide. And it turned out to be a peptide that started right here at the N-terminal end. And when he sequenced this, what he found then is the sixth amino acid in was a valine instead of a glutamic acid. So this is the guy way back in 1949, based on his analysis of peptide fragments from protease digestion of hemoglobin, that this was the molecular basis for sickle cell anemia. So the interesting thing to me is the first molecular disease. This was announced by Linus Pauling then back in 1949, 1950. He started using that phrase. And it has taken us 70 years then to actually come up with an effective treatment for sickle cell anemia. And the Obando story that Fran and Mark told earlier is just, it's an amazing story as to how effective this treatment is. So I want to talk, I think maybe there's one more slide here. Is there one more slide?
Yes.
Okay.
There we go.
Okay, so yeah, we're gonna switch to something else for just a second. How are you gonna get your kids? I shouldn't be saying this because I've never been in a classroom like Fran and Mark, so I don't really know how you engage high school kids in this topic. But But here's something that makes sense to me. First thing you have to do is get kids to think at the molecular level. And of course, kids have lots of experience in the macroscopic world. They know that if they cut themselves, they will bleed. And they know the blood is red. And you'll have to tell them that the reason blood is red is because of this heme group in hemoglobin. This is what imparts that red color to blood. And in particular, it's not just blood, it's the main component of blood are these little biconcave cells called red blood cells. And in every drop of blood that kids see at the macroscopic level, there are 5 million red blood cells in a drop of blood. So this micrograph here, under the microscope, you can see these red blood cells. So this is the microscopic level. And I like this photo because it gives kids the idea that, wow, there's a lot of red blood cells in my blood. But then David Goodsell has gone one step further because David Goodsell has painted a landscape of what you would see at the molecular level now if you were to cut open a red blood cell and look inside it. So what you see, all of those red things in the landscape to the right, each one of those little red tetramers is a hemoglobin protein. And here's a good number for you to know. There are 270 million hemoglobin proteins inside of every red blood cell. So I'd like you to try to get your kids to appreciate how many red blood cells there are in blood. And there's a number you can find out there on the internet. I think it's something like 15 trillion total red blood cells in your body. I don't know what that's good for, but inside every one of those very, very many red blood cells, there are 270 million hemoglobin proteins. So what do we use hemoglobin for? We use it to transport oxygen around our bodies. So you might ask your students to take a deep breath and hold it. So they breathe in, and then while they're holding their breath, you say, now this oxygen is flooding into your lungs, and your heart is pumping blood through your lungs. And as the blood goes through your lungs, all of these hemoglobin proteins inside of all of those red blood cells, they're loading up with oxygen. The oxygen actually diffuses very easily through the tissue in your lungs and into a red blood cell and loads onto not one, but 4 different heme groups contained in each one of the 4 globin chains in hemoglobin. So you can load an awful lot of oxygen up into one red blood cell. You've got a lot of red blood cells there. So after you've taken that deep breath and your kids are still holding their breath, waiting for you to tell them that they can now exhale, but you never tell them they can exhale. You just let them suffer. But then your heart will beat a couple more times and it will pump this oxygenated red blood cells out of your lungs, and it starts traveling in all over your body, delivering oxygen to your tissues where you need that to support oxidative metabolism. You need it to make ATP. You need it to be alive, okay? So that's one way I would try to get kids to appreciate how important it is that we have hemoglobin to transport oxygen. And then what a serious problem this is in people with sickle cell anemia who have a 1 amino acid change in their beta globin protein so that it distorts the shape of the red blood cells and it clogs up capillary beds and leads to all of these dire consequences. So this is just one more slide. I don't even know if we need to do this because Fran or Mark, someone showed this very, very well. So let's move right on then to not the Helen Obando story, but it could be. I don't think this paper that appeared in December of 2020. So that was like a month ago, right? So here's the paper. It appeared in New England Journal of Medicine, and the title said that CRISPR-Cas9 gene editing for sickle cell disease and beta thalassemia. So this is a report the reports on only 2 patients. One of them had sickle cell and one of them had beta thalassemia. So let me just talk about beta thalassemia for a second. That is a hemoglobinopathy. That is a disease of blood in which people don't make enough beta globin. They make the normal amount of alpha globin subunits, but they don't make enough, hardly any, beta globin. Therefore, they can't assemble hemoglobin tetramers, and their whole ability to transport oxygen is very much compromised. And really, the only effective treatment for these people is they are often in the hospital getting blood transfusions, like every 1 or 2 months. I think they're back in the hospital getting a fresh supply of red blood cells. So it's a pretty limiting life. All right, so here's the data, and if I could have the mouse again, because this is something that came up that I think we really need to talk about. In this panel right over here to the...
Pull your cursor to the left. Keep going. Here, I can point.
Yeah, we've got 2 screens set up here, you know, and the mouse goes from one screen to the other, and I want it to be over there. Yeah, all right. So this shows the synthesis of both alpha and beta globin, starting with the beginning of a pregnancy to birth, and then a couple of months after that. So in the beginning, when that fetus starts growing, the alpha globin chain gets turned on, and you start making alpha globin. But look at the beta globin chain then. At 6 months in, you're not making any beta globin. So you need hemoglobin in that developing fetus, but you're not making adult beta globin. You're making another form that's labeled gamma here, and we're just going to call that other form fetal hemoglobin right now. This is the fetal beta globin chain. So the fetus is forming their hemoglobin using adult alpha and a fetal beta. And then just at birth, right there, right at birth, the gene expressing the fetal globin gene gets turned off. And that's when the expression of the adult beta globin turns on. So at birth, you have this developmental switch from fetal to adult beta. And the mutation in sickle cell anemia is in the adult beta globin protein, the gene for the adult beta globin protein. Now, I should also mention that people knew that there are some individuals walking around out there who have what is called hereditary persistence of fetal hemoglobin. And for some reason, these people have a mutation in their DNA, which results in that fetal gene not being turned off at birth. It persists into adulthood. And these people seem to be just fine. And you know, if when you come to a summer course at CBM and we have time to sit around and talk about these things in more detail than we do right here, we can talk about those people and why they don't experience any problems in their adulthood. But here's the kicker. So if I don't see my...
It's over on the shelf there.
My gene sequence. Yeah, okay. I don't really need this model, but now, you know, this is a DNA sequence, right? And let's pretend for a minute that this is the beta globin gene, and somewhere in here there's a GAG that gets changed to a GTG right here. Turn this over. Here's a GTG. So that's the sickle cell mutation. So we want to change that. So you might think that I'm going to tell you a story about how CRISPR-Cas9, which is this protein right here, you might think I'm going to tell you how CRISPR-Cas9 edits this gene, edits the sickle cell, the adult beta globin gene to fix that mutation. And I'm not, because that's not what was done for Helen. Okay, and I should say, I'm not even going to say anything more about CRISPR-Cas9 because that's the subject of an entire webinar in March. So stay tuned for a later webinar and we'll talk about CRISPR-Cas9 does its thing. But can we back up one more slide?
Yes.
Yeah. And can I have the mouse? Can you keep the mouse over there? Yeah, okay.
There you go.
Okay, I think. Oh, why can I not do that?
What do you want me to point?
Okay, all right. So here you see CRISPR-Cas9 sitting up here, and it's binding to a gene. And you know, CRISPR-Cas9, the main thing about this protein is that it can target a unique sequence in the human genome. So there's about a 16 to 20 nucleotide long sequence that represents the beta globin gene. So we can, in fact, program CRISPR-Cas9 with a piece of RNA that will make it bind to and put a double-stranded cut in the beta globin gene, and that would inactivate it. But that wouldn't do us any good, just to inactivate a defective beta globin gene. So the story I'm going to tell you is a more complicated one. And if you look right down here, where did that mouse go? There it is. Okay, I hope you can see my mouse moving around. So there's something down here. It looks like this CRISPR-Cas9 is targeting a BCL, I don't know if it's BCL2A or BCL11A, but the BCLA gene. And what people have figured out over the years is that the reason this fetal gene gets turned off at birth is because this protein, BCL, is a repressor. So the BCL gets turned on right at birth, and it turns off then the fetal gene. So what they're doing for the Obando sisters, or Ellen, and also for the patient that they describe here, is they're simply reactivating the fetal gene, which is a perfectly good beta globin, fetal beta globin. They're reactivating it in Helen and the patient described in this paper. And those people are continuing to make a defective adult beta, but you'll see in just a minute in the next slide that that following this treatment with the CRISPR-Cas9, if you look first at the person with beta thalassemia, this is at treatment. They were making just a very little bit of fetal beta globin, but after the treatment, look at these blue bars that stack up here. This is the fetal beta globin. So 18 months later, you're making a whole bunch of fetal beta globin protein. Same thing happens over here with the individual with sickle cell anemia. You're not making much fetal globin in the beginning, but very soon after this treatment, you start to see the fetal beta globin gene get turned on. And then most importantly, if we go to this slide, this shows what happens to patients, these two patients following this treatment. Here's the patient with thalassemia, and each one of these little dots represents a hospitalization in which the person with thalassemia was getting a blood transfusion. So this is 2.4 years prior to the treatment right here. And then look what happens after the treatment. No more blood transfusions. So again, this is just amazing. If you listen to the stories about what a difficult life these people have. Here's the sickle cell patient, and here are hospitalizations for various crises. These are not hospitalizations, but these are just pain crises, vaso-occlusion events that these patients experience that knock them out of school for weeks at a time, and they're always struggling to catch up and behind. And again, it's a very stressful life. And here's when the treatment occurred, and then following that for 16.6 months, nothing, no episodes of pain crises, no hospitalizations. So this treatment, this gene editing, not directly correcting that one base change, that will happen at some point in the not too distant future. And we'll talk about that in March when we talk about CRISPR-based gene editing. But the easier thing to do, and the thing that has been done now for not only Helen and these other 2 patients reported in this paper, but I don't know, this work is going ahead so fast that it's hard to keep current with this. And I'm sure there are many other stories out there if we only knew about them. But it's an absolutely incredible thing that modern science has been able to do to finally correct, after 70 years, the first molecular disease. So it's a fun story to tell your kids, and it's a very uplifting one, especially if they've watched the Helen Obando video. They'll all. All right.
Okay, thank you. You are out of time.
I'm out of time. Right?
We are going to stay on and do questions after this closing. I just wanted to say a couple of things.
Can I say one thing?
Sure.
One of the things we haven't discussed yet that I hope we can in a follow-on question is this diphosphoglycerate and the affinity of fetal globin for oxygen as opposed to adult globin. So if you're curious about that, hang on for the question and answer session.
Yeah. Okay. And I have some of the questions up here, so I'll let you read those while I do our closing.
All right.
So thank you again to Fran and Mark for a great session. And I will just go over a couple of things. And if you have to go, that is fine. So Diane's posted the certificate of participation registration. So please click on that and I will send your certificate tomorrow or Friday. And then during the session, Mark and Fran used both that flow of genetic information kit, the classroom kit, and the nucleotide student modeling pack today. Tim used the nylon hemoglobin model, and he showed you our little beta globin mini model. And maybe we'll have that big plastic one available soon. Use the discount code webinar20 for 20% off most of 3DMD's kits. And watch your email tomorrow for our stock up sale on sickle cell disease associated products. We are offering up to 25% off when you buy 4 or more of our products, which include beta globin folding kits, the beta globin gene maps, mini models, and or hemoglobin poster. So we have a lot of other materials that will support you in teaching this story to your students. We have a break next week, but I hope that you'll join us again on February 10th when Karen Avery and Dan Williams present synapses. And please encourage your colleagues to register and join us. Please complete the survey that you encounter as you leave the webinar. We really do appreciate that feedback. And that concludes our formal presentation for tonight. Thank you for joining us. We'll continue to answer questions as long as we can and as long as you guys are asking. So this is the point where raise your hand and we'll bring you to the stage. And you can ask your question or go ahead and keep posting them in the question and answer.
So Karen put a question in the chat. Tim, why does the sickle cell HB stick together only under low oxygen conditions?
That's a difficult question to answer, and I can't give you a good answer. I see another question here wants to know the difference between oxygenated and deoxygenated. And I can't give you a good answer because it's subtle and yet it's real. So we do have models of oxygenated hemoglobin versus deoxygenated, and they look almost identical. The only way you can really see a difference is if you put some green struts between 2 different pieces. And in one case, oxygenated, you have a long green strut that becomes a short green strut. Come to the CBM, we'll get out those 2 hemoglobin models so that we can look closely at them. And perhaps we can offer some suggestion as to why that is. But I can't really give an effective answer to that question. Can I talk about the oxygen affinity of fetal globin? I haven't had time to read all these questions.
Yeah, so
Yeah,
I think
right here.
people had asked. that.
Susan Park says, doesn't the fetal hemoglobin have a different oxygen saturation curve than the adult? How does this affect the patient receiving therapy? Well, we're not going to talk about how it affects the patient receiving therapy. Again, come to a summer course and we'll talk about it. But I am going to discuss the first part of this because this is fascinating. This is also complicated. So I'm holding this hemoglobin model here. And if you could, again, you really need to have these things in your hands to really appreciate the power of these models. And I'm looking right down on it here, and I can see this space between the two beta globin sequences. And as someone said earlier, Fran said earlier, there is a small molecule called diphosphoglycerate. And all of our adult hemoglobin has a diphosphoglycerate bound right here between these two beta chains. The diphosphoglycerate is a very common metabolite to have in all of our cells, especially red blood cells. So those 270 million copies of this protein in a red blood cell will all have a diphosphoglycerate right here. And the strange thing is the effect of binding diphosphoglycerate right here to adult hemoglobin is to lower its oxygen affinity so that it has just the right affinity for oxygen, that it can pick up oxygen in the lungs and then it can release oxygen when it gets to the extremities where the oxygen concentration is low. So the diphosphoglycerate reduces the binding affinity so that it can release that oxygen in the extremities. Now, think if you're a fetus growing in a human, you're competing for the oxygen with the adult maternal hemoglobin, right? The red blood cells need to deliver oxygen to the fetus, but you need a fetal hemoglobin, which has a higher affinity for oxygen than adult maternal hemoglobin. So if the diphosphoglycerate that's on the maternal hemoglobin is lower in the oxygen affinity, all we have to do is evolve a fetal version of the beta globin, which is missing some two positively charged side chains right here in this cleft between the two globins, beta globins. Diphosphoglycerate is a negatively charged molecule. So there are two positively charged side chains that are missing, that are not present in the fetal beta globin, and therefore fetal hemoglobin does not have the diphosphoglycerate, which lowers its oxygen affinity. So it has a higher oxygen affinity than the maternal hemoglobin, which has the modulating diphosphoglycerate that reduces its oxygen affinity. So that's why we have evolved this strange developmental switch, because prior to birth, the fetus needs an advantage in stealing oxygen from mom. But once birth happens, then you start to become an adult right away and you're not competing with mom anymore. So then you kick in the normal adult beta globin. So,
and we have Jen Schrader on, and she has a teacher tip. Jen, if you're ready, you can probably go ahead and turn on your camera.
yes.
Hi.
Hi.
And I hope I can share a screen. Oh, can I share my screen?
Let me allow it.
An option. Otherwise, I can show you in paper.
You can share your screen also.
Yeah, we know, Jen.
So I took the model, so I just have the student version, and I was not wanting it to be three-dimensional when I stored it. So when I cut it out, I left a tab on the bridge, then cut a little slot. Hard to see. Cut a little slot right here where my finger is going. So you can slide it in and get the three-dimensional bit, but when you want to store it, you just pull that out and make it flat.
Oh, that's a great
Still
idea.
tape it on the green, but I didn't tape it on the blue.
Are you looking for a job? You should come and.
I am hoping that my school will let me have a sabbatical so I can come work for you guys for 6 months. So yeah, well, I'll keep you posted.
That would be great.
And I sent the screen. Let me get the right one.
Yeah. I received your email.
Yeah, so I just have too many windows open right now. So this is what I sent. If you guys wanted to be able to share that with others.
Perfect. I will definitely share that with people. We're going to move on to the next question. So Michael was asking, after the gene insertion for the fetal hemoglobin, will Helen be able to pass that on to her children? And will there be other consequences for positive generations for them?
So the easy answer to the first question is no, because there's been no manipulation of germline cells. So this is a Cas9, CRISPR-Cas9 treatment of immature red blood cells. So so the answer is no. It only will affect Helen, and it will not be passed on to her offspring.
Tim, can I interject something?
Yes, go ahead.
She did, so before Helen went under treatment, she did have an ovary removed. You want me to start? Oh, she did have an ovary removed and stored so that she will be able to have children someday.
Yeah, okay.
But yeah, so therefore, she still has a risk of passing on sickle cell.
Well, okay, I'm not sure she has much of a risk because
or I misspoke. Go ahead, Tim.
I see what you're saying. Yes. I kind of misinterpreted the question. Her kids are at risk of having sickle cell because yes, her germline cells have that mutation in the adult beta globin gene. Helen will be fine for the rest of her life, but it has not fixed or cured her germline cells. Now, in most cases, people consider that to be good because then people get real nervous when you start talking about editing germline cells. There are big ethical questions that come along. The other reason that sickle cell and blood diseases of all types are easier to treat is that you can, in fact, take some blood cells from a person. So this gets away from the need to match. You don't need to find a donor who can provide cells that are compatible with the patient. You just take the patient's red blood cells and you manipulate them out here in a petri dish and grow them up and check to make sure that you've gotten the modification that you were hoping for. And then you grow them up and you reinfuse them into the patient and you eliminate all possible graft versus host disease if you have a poorly matched donor providing some cells back to the patient. So.
Great, we still have lots of questions, and please raise your hand if you want to come up and talk to us.
Yeah.
We did have one question about the blue proteins. Do you know what these blue proteins are?
I can't name them.
Or do you know what they do?
Yeah, I can tell you what they do because I asked David about them just about a week ago.
Move these out of the way. Sorry, guys.
So, this is the great thing about David's landscapes because they're not just pretty pictures. They really have a lot of science in them. So if David were here, he would tell you exactly what those proteins are. All I can tell you is that the fibrous ones that kind of lie, parallel to the membrane, that's lamin. And that's a fibrous protein that interacts with those other purple proteins that are embedded in the red cell membrane. So all of those proteins are known, and they are known to contribute to this nice biconcave shape of a red blood cell. So all I can tell you is check out David Goodsel's website, and usually David publishes a paper in which he discusses each one of his landscapes. I don't know that he's done that here. but maybe you will find this in David's book called The Machinery of Life. I don't remember if he has a red blood cell in that book or not, but
we'll have to look.
Yeah, I'll find out what those are, and yeah, send me an email, and I'll give you the names in a day or two. Can I say one more thing about that?
Yes.
Can you show that red blood cell membrane again?
Okay.
So for those of you who were here, was it last week we talked about aquaporin?
Yes.
All right. So what protein should David have put in his cellular landscape? The question is, where is aquaporin?
Mm-hmm.
Because remember, Peter Agre won a Nobel Prize for finding the water channel aquaporin, which he purified from red blood cell membranes. So we're going to have to scold David if he did not put an aquaporin in that model.
Okay, so we have another question here from Rachel, and then we'll go to Gabrielle. So it said something on the slide bar graph, the blue is representing the fetal hemoglobin. What was the purple representing?
I don't know, you know.
If we look, so I'm trying to see.
There's so much good science going on here that I barely have time to read. I never read an entire paper. I was very happy to see what was happening with the blue bars.
You just looked at the figures
Yeah.
and skipped everything else.
I hardly even read the caption here. So it looks to me like that's called hemoglobin A2. And you know, here again, then we start to get into complications. So what in the world is hemoglobin A2? And I can't tell you right.
So I'm just going back and looking at this other figure to see if it's labeled.
No.
So hopefully we will share that link to that paper, and then it looks like you guys may have some homework for that.
So, you know, you can always blame it on someone else. A hematologist would know the answer to this question, but none of us are hematologists. So if there's a hematologist out there in the audience, please provide the answer.
Gabrielle, go ahead.
Yeah, because I put my question in the chat, in the Q&A, but. So,
yeah, I hadn't gotten to it. I was just about to, but then I saw
okay. So it sounds like the therapy of case 9 is to knock out the repressor for fetal hemoglobin,
yes,
so that the fetal hemoglobin will now be produced.
Absolutely right, yes.
But don't you still have to knock out the faulty sickle gene, because otherwise you may still sickle?
You would think the answer would be, yeah, we have to get rid of the bad protein. But in reality, you don't. What people find is that if you can provide, you know, I don't know what the number is, but say if 25% of the beta globin in an individual is fetal, then you dilute the effect of the bad beta globin gene. So you may still make some hemoglobins that occasionally have a beta globin chain that has the valine in place of glutamic acid. But that's not a heavy enough dose of that hydrophobicity that you start aggregating the hemoglobin according to the shape of the cells. Now, you know, if a student asked that question, I would pat them on the back and congratulate them for thinking, and because that's a very, very logical question. And the only answer I can give is that experience seems to indicate that you don't have to get rid of all of the bad beta globin protein.
I'm wondering if this ties into Mary's question. Does the alpha hemoglobin have a greater affinity for the fetal hemoglobin? So you know, that it's going to make hemoglobin with that fetal more than with the beta? Is that the question?
My answer is I'm not a hematologist, so I don't know the answer to that. I would guess not, but the true answer is I don't know. This is why we need to get those darn researchers involved in these conversations so that they can answer some of these questions with authority.
Okay, and we also posted, there is a JML tutorial that explains some of the structure things that you'll be, that's on the CBM website. So we'll make sure we share that as well. And then Jackie, we're reading your question as we go now. Speaking of ethical concerns, I'm surprised that the pre-implantation genetic testing is allowed to be done to have a child to help another child. Are there bioethicists expressing concerns?
Yeah, you know, I also had the same thought when I heard Fran talking about that. And, you know, I remember hearing that in one of the videos I watched. So yeah, they actually were trying to have another child for the expressed purpose, the hope of that child being a donor for Helen. And I don't know, I would have some ethical concerns, but you have to think about this family, because the son who was a donor for the older daughter, you know, he felt terrible. The whole family felt terrible that the older daughter, you know, got the cure, but that Helen didn't have a match in the family. So I can certainly understand why the family, my mom and dad, would want to have another child, you know, in the hope that they could produce another child who would be an effective donor for Helen, just as their son had been a donor for the older sister. Yeah, ethical questions, absolutely. But I can sort of understand why they would want to do that as well. That's a tough question. That's a tough decision for parents to have to deal with.
I think we're coming to the end of our questions. So if anything else has popped up, please raise your hand or post it. We do have one more about, is there any way to model mRNA editing? I don't quite, I don't fully understand.
Well, mRNA editing is again, it's one of these new things that people have now noticed is happening. I don't know the details of many of the stories, but as I understand mRNA editing, there are certain messenger RNAs that once they're transcribed from the DNA, from the gene, the nucleotide sequences of some mRNAs are changed, and it's only when they are changed that they then create a functional protein. Now that sounds crazy. You have to have some way of restricting those edits only to certain mRNAs and not all of them. So suddenly our whole central dogma would go haywire. So I don't know a lot about mRNA editing.
This was actually a part of one of the protein stories that we told, looking at spinal muscular atrophy and survival of motor neuron.
So this is a smart team.
This is a smart team with Mark McNally. So,
okay.
Right. So, Dr.
Go ahead, Mark.
Dr. McNally works with viruses and is spending his time looking at almost entirely RNA stuff. And he became really, really interested in these proteins called spliceosomes and how they work. And they're really, really complicated. There's 7 different gemin proteins that make this ring, and they're interacting with all this other stuff.
You're waving your hand.
It's like herding cats. Like it's crazy. Yeah. And so we were in the process of like trying to get students to wrap their brains around what was going on and how this actually happened. So we started talking about splicing factors and how there's tiny little proteins that bind RNA in specific spots. And we like, these are the green ones that say like, this is good, we're keeping this, and here's red ones, and these are bad, and these have to go. And how like a spliceosome comes along, it finds one of these spots, it actually cuts out a chunk, that's trash, and then goes down, finds the next spot, like here's stuff to save, here's stuff that goes, and then paste those things together before we send it out to go make a protein. And the other thing that we start talking about is the fact that like, we're always like, oh, hey, this gene codes for this protein. And that's like, that's true, but it's not always true. Like some genes code for lots and lots of different proteins. And this alternate splicing is one of the ways that we can take one genetic sequence and end up with thousands of different proteins. So it's like, it's a really, really complicated story. And like, we just scratched the surface of things that like we could do with it. But to help them understand what was going on, we took like little green flags and little red flags, and we put them on the RNA so that they could see, hey, parts of this are gone, and we have a new sequence of codons, and as a result of that, we'd have a new sequence of amino acids.
So what Mark is talking about is a student modeling program that's either known as SMART teams or MAPS teams. I'm not sure. This is probably a SMART team when you did that. But SMART is an acronym for Students Modeling a Research Topic. MAPS is modeling a protein story. And in both cases, these teams of high school kids led by their teacher are introduced to a local researcher, and they learn about the research that's going on in that lab. And they model a protein that plays a prominent role in that research. And they get involved in some really complicated, esoteric topics like this alternative splicing or RNA editing. And it's amazing what high school kids can understand and get excited about when they have an opportunity to start with something simple like the flow of genetic information kit, but then take those generalized principles and start listening to a researcher saying, yeah, that usually happens, but not always. So look at this cool science that we're just uncovering right now. So it's a great way to get kids to understand what research is and how science gets done.
Tim, Sue Holt mentioned that the Gritzel poster was done in 2005.
Okay, of hemoglobin.
Yes, I believe that's what she's referring to.
That sounds about right. Is it in the book? Do we know? Is it in Machinery of Life?
Yes, it is. That's why I raised my hand.
That's Kaylee, right?
Yeah.
He's like,
okay.
Oh,
look at
great.
that. Those blue proteins. Tell us right now.
So some of them, there are two types of blue shading apparently.
Yeah.
And depending on it, it is an antioxidant protein and it's a catalase.
Catalase.
I tell them what the.
I'm surprised.
Oh, and I was talking in our advanced molecular class about the different filaments. That fiber at the thing is actually spectrin. It's technically a different distinction of filament, and I'm sorry, but I have to say that.
Don't be sorry.
I could have sworn that was called laminin.
It's similar in function. They're just two different proteins.
Thank you, Kaylee, for
I love that DNA models on your mantle behind you.
There's mom's and there's mine. But I also had one more thing. It might be of interest to some of the teachers. But in my class, we talked about gene families and that hemoglobin is a huge gene family. I wonder if I can get closer. So you find that there's actually two versions of fetal hemoglobin, and that alpha and beta have duplicated and have technically differentiated over time. And I just thought that was really interesting.
Wow. And you can see, this poster here, Life is Complicated. So, we were talking about one fetal beta gene or one fetal, but you can see there's gamma 1, there's gamma 2, there's an embryonic fetal beta, embryonic beta globin. So you have to be careful that you don't get swallowed up by all the complexity. You especially have to be careful when you're just introducing high school kids to some of these ideas. So you want to be gentle. Remember the children, right? Don't confuse them. Don't scare them away from science. But at the same time, be ready to challenge those who always are pushing you to go a little bit further. So
we did have another helpful comment that the HBS abbreviation in some of those slides is the mutant sickle hemoglobin. Yeah. So
no.
Thank you for that. And so there's still 20, some of you hanging around. So please raise your hand and come up on the stage and say hello. There are so many of you who we've missed seeing in the last year or so. We'd love to talk to you. And if we don't get any takers in the next couple of minutes or other questions, we will go ahead and shut things down for tonight.
Yeah, and
When Fran was presenting, it made me think about our chromosome connections kit and the Punnett square, and that's based on the sickle cell mutation. And that's in our teacher resources. And so you can go in there and print the Punnett square.
That'll be coming in a future webinar. Is that correct?
Yes.
Yes, that's coming up.
February.
But the teacher resources, nearly everything is free. And so take advantage of that. And any of the PDFs you can print and use in any way that works for your class.
Okay.
So thank you, everyone.
Ooh.
It's great to see you, Jackie and Pringy and Mary.
Yeah, Liliana says hello.
Natalie, thank you.
Yeah, great.